Thanks a lot, Siva. Thanks a lot for your uh, very informative talk. Waiting for the question, I just want to asking about the software we can install locally and we can run it locally, or we need to upload the data into cloud in order to
Yes.
do the analysis.
Yeah, that's a very good question, actually. So there are two softwares which I presented. The CLC, let me share my screen again.
Um,
Okay. Can you see my screen?
yes, I can see you right now.
Yeah. So that's a very good question, actually. The CLC, the software which I mentioned, yeah, here, this slide. So the CLC is a local software. It runs locally where you provide a fast queue. Either it runs on your local desktop or server or on the things. And the VCF which generates, you have to upload it to our server, which is present in Australia or in US. Okay. So that we have it as a lock and key model where we lock it to your own system. Like when we give access to you to use this QCI, we make sure that only your information is visible to you. Okay. Even
Mhm,
I or any other colleagues from Kaizen won't be able to see that. So in that
mhm.
way, we have the HIPAA and data security compliances afterward. So QCI, you to goes to the knowledge base which is sitting on the VCF file, goes to the knowledge base which is sitting on the server, brings the relevant information in the reportable format. That's how it is used.
Okay. So,
Yeah.
and then my next question concerning about the configuration of my computer in local in order to can run the CLC. Uh,
Yeah.
how many CPU do I need? How many RAM memory do I need? And how many storage do I need?
So yeah, that's also a very good question on the point of how many samples you are doing. So per month,
Okay.
how many samples turnout you are doing? That's one thing. How big is your data? Are you running a small panels? How much GB? Whether you are converting 100x, 200x into 2 GB, 3 GB data.
Okay.
So
Okay.
how much complexity is there? So those things decides on how much usage you need to do it. Okay. So those are the steps which tells you if any small panels with 16 GB RAM and 500 GB to 1 terabyte of hard disk, it runs very smoothly and it can run fast also. Okay.
Mm-hmm.
And our tools are ready to support whole genome and whole exome also, provided you run it on the capabilities. Also, you can upgrade it to, that is why like we go to server or in the uh, cloud module to support this. Okay.
Okay. Okay. And then the VCF file, do we need to
Mhm.
do the join call in order to have one VCF file for each run? For example, each run we have 48 sample per run. And then we need to do the join call to have the one VCF file containing 48 sample. Or we submit or we upload individual sample in the VCF file into the
Yes.
cloud.
So actually, there are two ways also. So when you upload
Mhm.
to QCI, you can submit as like all the different files. You can submit as single file also. If you can join it outside and do it as a single file uploading, or you bring individual five VCF files for a single patient and you upload them as a same patient sample. So you can merge
Okay.
it within QCI itself. So that option is
Okay.
also available.
Okay, okay. So you
Yeah.
said that in the nearly the last line, you said that you are waiting for the IVD for the software. So the IVD for the cancer diagnostic only, or
Yes. Yes.
for
Yes.
every
Cancer
job
only.
line, or for
Yes.
cancer only. Okay.
Yes, yes. We already have, see, we have IVDR certified in Europe. Okay. We, as
Mm-hmm.
per the, like, as I think Tanat was also mentioning, each country has different rules and regulations. And
Mm-hmm.
we have compliances of Europe. We have followed it. And we have got this QCI, especially for somatic, has got the IVR certifications in Europe.
Mm-hmm. So
Okay.
then um the variant annotation for the somatic is follow the ACMG, or you follow what kind of guideline?
Yeah. So actually, there are two things. uh, uh, for any somatic variant interpretation, there are two things. Okay, 1 second. Yeah, you can see, right? Actionability means what are the drugs approved, whether there is a approved drug available, whether there is a clinical trial. So that is called as actionability. That is provided for as per American
Mm-hmm.
Molecular Pathology or CAP or ASCO guidelines. Okay, the
Mm-hmm.
pathogenicity is standard as per the ACMG guidelines. So we
Okay.
get both of these for this variants.
Okay. Perfect. Yeah. Thank you. So and then we have the question from Dr. Tan. Please directly asking, turn on your microphone, please.
Yeah, thank you very much for your presentation. And I have a couple of questions about the
Yeah, please.
criteria for variant report and some questions for actionability criteria. For
Okay.
for variant report, I would like to know what is the threshold for the the depth of sequencing and also the thresholds for VAF. Uh, I saw that in your presentation. There are variants with only more than 100 read more. Yeah, the depth is about 150x. And is it too low to report for somatic samples? This is
Okay.
my first question, and I will ask just the following questions
Yeah.
after.
Sure. So thank you, Dr. Tek, for this question. But so technically, QCI is just an interpretation of the information. Okay, the one which decides the variant allele frequency (VAF) is through the secondary analysis tool, which is there like CLC Genomics Workbench. So we, based on the like your wet lab, you can have 100x coverage, and what is the gene which is mutant, which is coverage. We can go up to 0.5x also. Okay, 0.5% also, provided how much is your coverage. So if you have 500x coverage, we can go low as 0.5% also. Okay, but if you have only 100x, so see, 1% means out of 100 cell, one cell has that cancer mutation. So do you think like so our assay should be sensitive enough to detect that? So that's where we recommend you to run some of the positive control samples, okay, first for the assay validation, and have those mutations. Like there are some companies which provide you Horizon and other people, they provide you samples, cancer samples, which are known, having known mutation with particular allele frequency. So you run that and you standardize the wet lab, and you see whether it is same thing is detected at same allele frequency in the um, secondary analysis like CLC Genomics Workbench. And also I will tell you, if we compare between Illumina and Ion Torrent and long reads like ONT, there will be a lot of difference.
Okay, so is there any also criteria for sample selection before testing? I mean that tumor percentage, for example.
Yes,
Sometimes,
yes,
we have small samples with low tumor percentage, so it's
yes,
quite
yes.
difficult.
Yes, yes, yes. Definitely, there are some criteria like minimum, uh, so we say anything about 20% tumor content is always better to look for that, because it avoids the dominance of normal cells. And then also it is important to have the fresh, the what kind of samples. If you're looking from FFPE tissue, again, that's another challenge we have. So these contents also helpful for that. So tumor content as well as the tumor DNA quality. These things are also very important parameters to look into.
Okay, so thank you. And my next question is that I saw in your presentation you use AMP and CAP and ASCO guideline for classification of
Yes,
actionability.
yes.
But actually, your program gained the IVDR approval from the EU.
Mm-hmm.
Why in this case you don't apply the ESMO guideline? which is more, appropriate, which is more suitable for EU regulation in the classification of actionability? Because
Yeah.
yeah,
So
just continue, please.
agreed. No, that is a very nice notice, nice point you have caught up. So actually, the classifications or the guidelines we follow from AMP, CAP, and ASCO. But the if I show you here in the next slide, where is it? So we have all the information from NCCN to ESMO, NCCN, and other data like ESMO, NCCN. You can see here, right? ESMO is there. Okay, NCCN, PDA, and you can filter by that also. So it's there. So that's already part of it. It's the actionability criteria which are approved and followed worldwide is as per the guidelines what I told you, AMP, ASCO, and this one. Okay, and also currently we are in a transition phase. The whole world will go to VICC guideline system in coming next year. So we already providing customer VICC score and how it is helpful. So that is will also be launched. I think by this 2 year end, all the people will move to VICC and even QCI will adopt it. So that's how it will go. And just to answer your question in one line, yes, we have ESMO and everything. Only for the actionability perspective, the approved methodology is as per the AMP, ASCO, and CAP guidelines. So we have that standard guidelines used there.
Okay, thank you. So my last question is that for actionability criteria, classification. I saw that these are, for example, like 1A, 1B, 2A. So I mean that this is just a general combined classification provided by QGIS, or you based on some specific classification. For example, for ESMO, there is ECAP classification for actionability, and also for ASCO, there's another classification. So, just back to a couple of slides, please.
Yeah. Actually, that's what we take all the things. It's not just one thing. Okay. So even if it ESMO says this one, it will be reported there. Okay, and we leave it to the end user to choose whether they want to go with ESMO or ASCO or anything else. Okay, so all the relevant informations will be there. If it is classified as 1A and ESMO has agreed and somebody else has disagreed, so that information will also be there. So if you click on this small triangle, right, what different people like ESMO, ASCO, everyone has told, that will be there as listed, what each person has told for this variant.
Okay, thank you very much.
Okay, I hope it answers your question. Thank you, Dr. Ryan.
Thank you, Siva. Thank you, Dr. Thịnh. So we still have, so I let you take a break before several questions come to you, Siva. So
yeah, sure.
The next question, so in Vietnam, they do some kind of sequencing, and then do they need to submit or upload the data, the raw data, to Australian to have the final report?
No, that's what I was trying to tell you. You are not uploading.
It's not me. It's from the other one.
Yeah. So that's what the first part is. You have the raw data that is in FASTQ format. So that is you are not uploading it to any server or any cloud material. You are doing it locally on your laptop or on your own server. Okay. Then you are getting a VCF file. That is a file which has the variant from the mutation. Okay. But that doesn't have any patient information. But the thing is, the knowledge base means what is that clinical trials, ACMG, publications, everything is sitting in the server, which is there in the Australia or in US. So we have to protect that. So that is where we have a web-based tool that is QCI. Okay. You use QCI to upload your VCF file and pull out all the relevant information from the knowledge base, which helps you to classify whether there is an approved drug, approved therapy, approved clinical trials, what is happening in there. So that information. So that step 2, this is where it will go to a approved server. But we also have, that's why I mentioned to you in the last slide, it is well HIPAA, ISO and certified, and a lot of people use it globally. And we make sure that data protection policy is strictly followed. So we both use up the sign of the agreement, where, even if you ask me, I won't be able to look into your data until and unless you provide access to me. Okay. So that's how the lock and key model works with you. So when, like in your institute itself, not everyone can visualize. Only the ones you have mapped to that tool can only visualize those data and informations. And also, we don't use those data for anything else.
Yeah, thank you. So, bạn,
I hope this clarifies the answer. Yeah.
yes, em will translate into Vietnamese for him. Yeah. Bạn Nguyễn Hoàng Huỳnh
Yeah,
Anh
please,
Phúc
please.
thì có nghĩa là data của bạn phải có một cái máy đó gì đó local, cái máy ở văn phòng của bạn á, để bạn phải có cái cấu hình máy á, nó phải phù hợp với lại ờ, cái lượng data mà bạn giải á, để bạn có thể install cái software CLC đó, rồi bạn upload data của bạn lên vô trong cái CLC đó. Thì dĩ nhiên là cấu hình máy mình phải bự để mình đủ chạy, phải đủ lớn để đủ chạy cho cái data lớn của mình đó. Và dĩ nhiên là cài cái máy đó thì cài cái software COC thì phải trả tiền đó. Rồi xong xuôi rồi bạn ra được cái file VCF file, xong rồi cái VCF file nó sẽ chứa những cái biến thể á. Rồi bạn lại upload lên trên cloud, nó lại có một cái software trên cloud đó. Là như cái bạn Lưu Anh ở đây, tôi xin lỗi là tại tôi không biết bạn Lưu Anh là ở đâu, bởi vì tôi hỏi cái bạn Siva cho nó chắc ha. Thì bạn Lưu Anh là viết lại chính xác rồi đó, cái mà ờ, tiến sĩ Siva nói, được không anh Phúc? Em nắm được không em? Rồi, so so the next question about the software, you said that you have the IVD in Europe. It's not yet, but it coming soon. Yeah. And he asking that how about in Vietnam? We can using
Yeah.
it for the Ministry of Health to
Yeah.
satisfy the regulation from the Vietnamese Ministry of Health or not? Yeah. So the question.
See, yeah, thank you. That's a great, again, the looking at the perspective. So we had like each country wise, the rules and regulations are different. So that is where we are working on that. And also, see, there are every quarter we try to do some improvements. So we are working on that based on the number of samples. If more samples are there in each country, based on that we will take the priority. So we also have like in another few more months, we are trying to using AI convert into local languages also. Like Vietnamese, can you publish a report in Vietnamese? Something like that we are working out in backend. I won't tell that it will come this state, but we always working on new things. Yes, and we also want to have like a IVD for the entire thing, but only thing because it's limited, since we have different rules and different application forms itself to submit to each country, and their rules and regulations are different. It will take some time to prioritize and get that approval done. And also these are very, they are not constant, they keep changing. So that is another thing which we need to make sure everything is in right place. Okay. And majorly because of few big projects, we were able to do it in Europe as a priority.
Okay. So the next question is concerning the update of your database. How often you update your database?
Yeah, that's a important and relevant information, because this is where you want a quality data, and as well as content which is trustable and which is up to date. Okay. So last week, if ClinVar has released something or clinical trials has done something, you get that updated everything. Okay. Even the publications also. Okay. If you yourself has published some data and it was released last week, then we make sure that we get the updated content within the 2 weeks of time. As well as every quarter, we do some feature changes as well as all the informations. But ACMG or ASCO or anything criteria has changed. we try to make sure as soon as possible so that the latest information is got to the end users.
Yeah. And then how to deal with the inconsistent. Like for example, last year we advertised with the pathogenic, but this year we advertise with the lightly benign. And how come we can dealing with the situation?
Yeah. So that's also you can re-index the sample. Okay. So your samples, you just by clicking on the last year sample, it will rerun the sample and it will tell you last year it was likely pathogenic, but due to this evidence, I make sure that what is that evidence is clearly highlighted in some color so that it may upgrade your sample to likely pathogenic to pathogenic or tier 2B to 1A. So that also we provide you the information on that. Okay.
Okay. So the last thing concerning about the in silicon prediction tool, like the in the CLC software, they use the CARD, the CIDD score.
Yeah.
However, in some variant, it's not a binary score, and there is software UDR, the prediction tool, such as the SHIP or Polypen tool or Revel.
Yeah. So we have 11 prediction in silico prediction tools, which predicts whether this having this we have Reveal, we have CAT score, we have Splice AI, and we have QCI interpretation also, and we have expert interpretation team. See, there is an oncologist, 200 of them sitting in US and Europe and till to Japan. They curate the content and they also look through the evidences specifically from oncology perspective and report it. So all this together helps us to predict this. We have, just to answer, 11 in silico prediction tools which detects whether this having this mutation leads to gain or loss of mutation or leads to a deleterious protein or a gain of function. So this information is provided to you along with the expert interpretation also. So this is something which you won't get it anywhere else. Yeah.
So the tool is very divergent. And what is the response for each stone using? Where come from? You using the original recommendation, you follow up the ACMG or LinkedIn publication or whatever.
Yes. So we actually from the content wise, we look from the ACMG guidelines and the priority for pathogenicity and the ACAP ASCO guidelines for actionability, as well as the clinical research data which is there. We provide you that information also, and they are categorized as a different standard or moderate or strong support. Okay, so based on that, the criteria will change and the priorities will also change. Okay.
Yeah, thanks. Dr. Phúc Đinh, are you happy? Are you satisfied with the answer?
Vâng, em hài lòng với câu hỏi ạ.
Ok, câu trả lời á, câu trả lời là em happy rồi đúng không em?
Dạ vâng ạ, em happy với trả lời của tiến sĩ Silva.
Ok. Nếu mà cần em cứ hỏi thêm không sao đâu em. Đó, câu hỏi cuối rồi. Ok, so that's the last question. Thank God. Dr. Sita, like you have the oral exam.
No worries.
Thank
No,
God
this
for
is
your
because
time.
that's an important step. I will tell you, when we report the information, it should be benefit to the patient who is the suffering and we are all doing it from sample to clinicians to everybody is trying to help the patient. So it should go as a relevant information. I agree with all the questions what you have asked. Those are nice questions. Thank you.
Yes, because our student is more about clinical student, the medical student. Yeah, so that is in that the question is very specifically. Yeah.
Sure.
So, chỉ muốn nhắn nhủ với bạn Nguyễn Đoàn Huỳnh Anh Phúc á, là cái IVD này là chỉ cho cái ung thư thôi. Cái germline thì không có, được không em? Đó. Bởi vì là không phải cái IVD nào nó cũng là IVD cho cả cái software hết, mà nó chỉ có cái phần là cho cái phần report cho cái ung thư thôi, được không em? Đó. Hồi nãy tôi có hỏi á, lúc đó tôi có hỏi là cái IVD này là cho cái gì đó, bởi vậy là mình cẩn thận cái đó, chứ không phải IVD là nó universal, được không em? Đó mà người ta cũng chưa có nữa đó, người ta cũng đang chờ. Đó. Rồi, thanks a lot for three audience. And once again, all we together, we would like to say thank you for your time as well as your very nice talk and information. Yeah. Không biết các bạn có hỏi thêm gì không? Chung hết từ đầu đến cuối á. Có hỏi gì không mọi người? Rồi, không hỏi gì hết thì mọi người thấy là mình học đó, mọi người cũng ôn lại cái bài của mình ha. Đó, là mình học tách chiết rồi xong xuôi rồi tách chiết thì mọi người cũng biết cái mẫu, mỗi loại mẫu nó sẽ có khó khăn khác nhau mọi người. Đó. Ở đây mình làm là thực tế, đó mình muốn không phải dạy lý thuyết không. Đó là thực tế nó có như thế nào mọi người cũng thấy đó. Mình có tách chiết đủ loại mẫu hết trơn. Đó, mỗi cái nó có khó khăn riêng của nó. Đó, bởi vậy là mình giống như thông qua cái, tại vì cái này nhà người ta sản xuất mọi người, đó là người ta hiểu rõ nhất chứ không có ai hiểu rõ bằng hết. Tại vì người làm nghiên cứu thì chỉ tập trung vô nghiên cứu thôi, đúng không? Ở trong lâm sàng, trong thực tế mình đâu biết đâu. Đó, tôi không biết, tôi nói thiệt. Đó là phải điều kiện như thế nào. Đó, có những cái là học hỏi rất là nhiều. Đó, thông qua mấy cái buổi nói chuyện này thì mình mới hiểu rõ được. Đó, tại vì nhà sản xuất mà, người ta sản xuất ra cái gì thị trường nó cần, chứ không phải tôi nghiên cứu trong phòng thí nghiệm không có gì đó. Và nó có những cái chuẩn tôi cũng không hiểu, không biết nó là gì, đúng không? Đó, ví dụ như tôi làm nghiên cứu thì đâu có biết IVD nó là gì đâu, đúng không? Mọi người cũng thấy là bữa nay người ta cũng trình bày ra cho mọi người thấy. Đó. Rồi, nếu mà có hỏi gì thì mọi người cũng thấy là trên trển người ta cũng có đưa cái thông tin đó, cái địa chỉ để liên lạc rồi này, cái email đó, để mọi người có thể hỏi thêm nếu mà ngại hỏi ở đây ha. Một lần nữa cảm ơn các bạn tham gia cái lớp này. Đó. Ờ, và mấy tuần sau nữa thì cũng sẽ có mấy cái sản phẩm khác nhau nữa mọi người. Đó, cái nào nó cũng sẽ có ưu điểm có khuyết điểm đó, thì tùy theo cái nhu cầu mình đang làm cái gì thì cần cái gì thì mình sẽ tham gia để mình có kiến thức của mình để mình hiểu chung luôn mọi người. Đó, có cái nhìn tổng quát đó. Ví dụ như là tuần sau là sẽ có cái bên DKSH mà phân phối cái hệ thống PacBio là long read, đó là cái phần mà cuối cùng của cái bạn báo cáo thứ hai. Đó, cho thấy là PacBio ta cũng đang sử dụng để giải đó cho những cái sản phẩm dùng để chẩn đoán bệnh mấy bệnh di truyền, xét nghiệm gen ha. Đó. Đúng rồi, người ta bên Bắc Bio có long read và short read luôn nha mọi người, chứ không phải chỉ có long read không đâu ha. Cái short read của người ta là cho cái ung thư đó mọi người, tại vì nó rất là chính xác, nó rất là tốt luôn. Tôi phân tích mấy cái data thì Bắc Bio data là định B, nhìn rất thích mọi người. Đó. Again, thanks a lot for your time. Cảm ơn Kaya Trình nha. Thanks a lot Kaya Trình. Cảm ơn bạn Tùng rất là nhiều. Đó, đã hỗ trợ chúng tôi có một cái bài giảng rất là hay ha. Và bây giờ cũng tối rồi ha. Chúc mọi người có một tối ngon. Thanks a lot and good night for everyone. Good night Siva, Nathan and Chi. Good night.
Dạ, cảm ơn anh Lợi nhiều lắm ạ.